Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. We are very excited to bring you a new episode of the podcast. Before we dive into this one, we have a few announcements to make. The program of the upcoming third edition of the Dr. GPCR Summit is taking shape. This year, the meeting will be held between October 10th and 16th. The meeting will be virtual so that we can allow as many participants from around the world as possible. We will host live talks, which will be hosted on Zoom from October 10th to the 15th. Everyone is welcome to participate by either listening into the talks, presenting a poster, or submitting a pre-recorded talk. We will also have prizes for trainees for their great presentations. We are also trying out a few new things. We'll host networking and poster sessions for the first time on Wonder. We will have also a full day dedicated to trainees and to trainee talks. We'll be hosting also workshops on data analysis, on how to preserve data integrity in the lab, and how to use GPCR DB. Everyone is welcome to attend. And it's free when you become a Dr. GPCR Ecosystem site member, which is also free. Speaking of the ecosystem, we are excited to share that the Dr. GPCR Ecosystem 2.0 platform is now open. Visit drgpcr.com to explore the ecosystem. The ecosystem is your GPCR-focused virtual playground. Join over 50 of your peers who have already registered as site members. You'll also have the option to select a plan once you're a site member. You'll get access to all things Dr. GPCR, including access to new podcast episodes before they even get released to the general public. You'll get access to our new group discussion and forums, our job board, and our learning center, while you'll be able to take a course or even prepare and share a course of your own with your colleagues. You'll also be able to discover GPCR companies and much more. Take advantage of everything that the new GPCR dedicated online playground has to offer today. Once you're a site member, you can also become an ambassador, which means that you'll get your own dedicated Dr. GPCR Ecosystem affiliate link. And every time someone subscribes for the year to the ecosystem, you'll get compensated. This will potentially help you offset the cost of the yearly membership. Last but not least, we're also looking for content creators. Subscribe to the ecosystem and start writing your own GPCR-focused content, share it, and show off your GPCR talents. Visit drgpcr.com to find out more about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I have the pleasure of having with me Dr. Jean-Philippe Pin. Hi, Jean-Philippe. Nice to have you on.
Hello, Yamina. Nice meeting you again.
It's so great to have you on. And before we kind of hit record, we're just mentioning that we've met where we were at the same conference. I even remember sitting at the same table as you for breakfast at some point at that conference at the Keystone in Ireland. So great to have you on today.
Thank you.
All right, so let's start at the beginning. Would you please walk us through a 30,000 feet view of your career as to where you started and where you are today?
So it's an interesting journey in a way, because I started being a teenager fascinated by insects and plants, and this bring me to study biology. And my dream was to become a biology teacher. I didn't want to be a scientist. I really wanted to be a teacher to save time going into the countryside, looking for insects, birds, and flowers. And that's why I started to study biology. And I don't know if I would say lucky me, but I met very impressive teachers at the university. Philippe Pin is one, for example. And they really impressed me by what's going on in science, especially in neurobiology. And that's when I decided to turn off my mainstream from becoming a teacher and to go into more science and become a scientist. So then I started to study receptors, and especially glutamate receptors. This was in the early 80s, quite a while ago now, 40 years precisely. And I started to study glutamate receptors at a time where glutamate was not even recognized by anyone in the community as being a transmitter. Was still a question, how can such an amino acid be a very important neurotransmitter in our brain? And that's why I was studying glutamate receptors, or I would say excitatory amino acid receptors, possibly not being activated by glutamate, but possibly being activated by other kind of acidic molecules like cysteine sulfonic acids or other compounds that would be more specific to our human brain to be dedicated to such an important function. And that's how I get into the discovery of specific glutamate binding sites that were very different from the ionotropic receptors, the NMDA, AMPA, kainate receptors. And then we postulated, being in Joël Bokert's lab, Joël Bokert is well known in the GPCR field, that probably glutamate could activate G protein coupled receptors. And that's how, at the very early stages of my career, I get really lucky. This is luck. And science, you know, you have to be smart, you have to be lucky. And this has been a really very nice luck that I participated in this discovery of the so-called and well-known now metabotropic glutamate receptors. That's how everything started. And that's how I jumped into the glutamate receptor field. Mm-hmm. And then I went to do my postdoc in San Diego in Steve Heinemann's lab with the goal of cloning these famous mGlu receptors. I lost. Sometimes you are unlucky in science. And in that case, the group in Japan was much more successful than I was. And they cloned one receptor after the other. And I was, you know, trying to catch in that field. But it's been quite difficult to compete because I was alone in a ionotropic glutamate receptor lab, with all the technology, all the tools, but not necessarily all the brain inputs that could be useful for my project. But lucky me, when you have, you know, sometimes you bad luck, but you also have good luck. At the same time, I got a permanent position in France. So this was by the end of the 80s, and it was still possible to get a permanent position in France in the early stages of your career. So then I came back to France in '92 and set up my group working on metabotropic glutamate receptors. And we get from discoveries to discoveries that these receptors are so fascinating. And I had a very nice discussion with on Visio with Skiniotis, you probably know, who is now doing a lot of solving a lot of structures of class C GPCRs. And he discovered these receptors through the cryo-EM analysis, and he said, these receptors, these guys are really fascinating. And they are, and I'm still fascinated by these receptors, and we still work on them 40 years after I participated to their discovery.
That's so amazing. You made a couple of interesting points. So I'm going to take you back to the beginning of what you mentioned. You said as a teenager, you wanted to be a teacher. Do you have any teachers in the family? What drew you to teaching? I understand the biology aspect, the insects and the plants, but
it's true that my parents were teachers, so maybe this influenced me. But the good thing is that I realized that going into what you love, and also what I really wanted to do was to share what was really enthusiastic to me. And sharing what I love is something that is very important. That I keep still going this way nowadays in science, because when I teach, I really love teaching, and apparently the students also enjoy it. And sharing what you know is something that is essential. And also, you know, being a teacher, you have a lot of spare time. This is something that I was also looking for family reasons. You know, you have time to take care of your kids, you have time to take care of your home, and you have time to take care of what you really love. I mean, going into the nature and And so this was my goal when I was around 18 or something like that.
Did you I remember you mentioned biology, but were you good at other scientific topics like math or chemistry or things like that? Or it was just both?
No, I was fascinated by biology. I was fascinated by nature. But I was good at everything at school, for sure. So I was this kind of, pupil who had no big problems in the system, both in math and physics, in most scientific matters.
Yeah.
But not in French. So my French teacher, when I was at high school, told me you shouldn't go for long studies because if you become an engineer with a lot of responsibilities, you have to know how to write things. You have to know how to write reports. And it turned out that she was the sister of a researcher I met later on in my career. And so we had a discussion later on, and I told her, yeah, I was not good in writing in French about all this, 17th century poetry or whatever. I was not interested in that. So it's very difficult to write good reports if you don't love what you talk about. Yeah. But I think I'm pretty good in writing scientific articles on grant application and so on nowadays. So I was not that bad in writing, but as long as you write something you love, I think you can really express yourself.
Yeah, it's quite
something that is.
Yeah. That's so funny. What was her response? What she?
I was. This was when I was preparing my PhD. So her response was, "Well, okay, that's
good for you."
Good for you, and I missed the point at that time.
Yeah, no, I agree with you on the fact that if you do something and it comes out through what you're saying, is that you really love what you're doing, and it doesn't feel like work, and it's a pleasure doing it. And exactly. I feel the same way recording podcast episodes. It's effortless. It's just I come out energized. I'm excited to do these things. And I have to agree, I went to school in French as well in Canada. And yes, I did have French classical, literature courses. And sometimes it was very difficult to read through all those, stories, those books, and then write a report about something that actually happened at some point and they just don't care about.
Yeah.
And the French language.
And you can change your mind. Nowadays, I love reading these books, but when I was a teenager, I didn't.
You're right, because after I finished school and I didn't have classical French literature classes, I loved reading in French. And don't get me wrong, I think the French language is such a beautiful language, but at the same time, it's such a difficult language to learn if you're not immersed in it.
Yes, I guess.
So
I imagine it's much more difficult than English, especially because English is, used all over the planet, and probably Shakespeare is not very happy about how the English language became.
Probably not. But I have to admit, I wrote my master's and my PhD thesis in French, and I think the thickness of those would be like 1/3 if it were in English, because and I do that even now with French correspondence. If I write an email, I start out in French, and then the next sentence is, I'm sorry, I'm switching to English because it's going to be much faster for my brain to process. The second piece of information that you shared with us is the luck in science, and sometimes you're unlucky in science. How much do you think luck versus hard work contributed to your success? 50-50, 75-25?
Hard work for sure, but I never realized I was working hard. As you mentioned, I mean, if you're doing what you really love to do, at the end it's not working. So what I think it is really important is to be lucky, but also to realize you are lucky. And to realize you are lucky, you have to put your brain on the table, smash it, and think about what you observe. And then you realize that there is something exciting here, and it might be luck. And then you have to go on this. So I think luck is, something you have to realize, and to realize that you are in front of a lucky situation. It takes also a bit of your understanding of the situation, analyzing of the situation, and then concentrating on that situation and say, "Oh, I'm going to make a story out of this."
Yeah, and you make a good point in here as well, is really focusing on the big picture and trying to get that piece of information that you can, build your story on, and make potentially interesting discoveries for the field and in science in general.
Yeah.
Third interesting point that you made, you mentioned, being scooped and trying to clone these receptors during your postdoc, and you mentioned that you were working alone. And I think to that point, I think collaboration in science is absolutely crucial. Working alone is not an option because of the sheer volume of the work, but also the difficulties of the problem.
I think it was still possible in the 90s. You could do good science being alone. You could do a very nice, neuron paper just being an electrophysiologist and recording synaptic activity, whatever. This was still possible, but I guess nowadays it's impossible. Because if you look at the publications, high-profile papers, really good stories, you will see that you have different techniques that have to be put together in order to make a story. And by yourself, you cannot handle all these technologies. I mean, going from electrophysiology, immunostaining, cloning, threat studies, whatever, all these things. I mean, one person cannot, control all this. So you have to deal with people having different knowledge and different know-how, put them together in order for them to work on the same subject.
I think so too. And I think it's a
very
good
important.
thing. It's important, but it's also a good thing, because when you think about it from a holistic view and advancing science and understanding GPCR research or GPCR biology, it's good that we have all these techniques now. Mm-hmm. And it's also good that we can work together and pool all of these resources in order to answer higher level questions.
Yeah. One thing I want to make also that it's really nice to go out of your own field. And being a glutamate receptor guy, at the beginning, I was very well recognized in the glutamate receptor community, but not at all in the GPCR community. And then I get to know how people in the ionotropic glutamate receptor field were working. But at the same time, because I was interested in the GPCR, I was also attracted by the way the people from the GPCR were approaching things. And that's very interesting, because you will see that these two groups of people may ask the same questions, but may not approach them the same way. And you learn a lot from, being in contact with people from a different field.
I think you, again, another great point to really look outside the field, because sometimes methodologies, techniques, potential collaborators could really help advance the work.
Yeah, it's the same. I mean, I get some project on the tyrosine kinases receptors, and this field is, in a way, if you look at this field, it's going into cancer biology, basically. In most studies, they go into cell biology. But if you look at the structure, function, dynamics, dimerization, all that stuff, they don't approach these questions, whereas we do so in the GPCR field. Yep. So it's interesting to see how the fields approach the different questions. And some going to concentrate on something, another field will concentrate on a different questions, a different approach.
Great. And when I think about tyrosine kinases, in my mind, we know everything. It's done. There's nothing to do, but it's not true.
It's not true.
There's so much to be done. But just because, when you focus on one thing, which is GPCRs, then it's difficult to focus on other things. But I think you need to be able to pull out of your own field and look around and see what happens elsewhere. And is there anything we can bring in? It's similar. You mentioned cancer, and a lot of people don't think about, G proteins or GPCRs as being in cancers now, where the cancer field doesn't think about that. There are markers, but that's that.
Yeah, totally true. I mean, I don't know. We did apply 3 or 4 times to grant agencies in France in the cancer field, and we never, ever get funded. Even though I'm really sure that what we propose is really innovative, novel, and everything would have been a breakthrough. I mean, mGluRs in cancer, this is obvious. And we never get funded to work on this.
So, yeah, because
it will come out one day. I mean,
who knows? Who knows?
Yeah, for sure.
I think it's a matter of time, and it's just it's a matter of time, and it's also a matter of keeping at it. I think consistency is important, and not giving up is important, not only in the everyday lab work, and this is mainly to postdocs and students, but also at a higher level. When you have an idea, you have to follow through and go for it. I remember seeing Brian Kobilka give a talk, and I think this was prior to the Nobel Prize, and he was mentioning that he was almost out of funding, and he had these purification columns in his office, and he was almost, I think, putting in his own money in order to continue and do this work, and he persevered. And once he got the Nobel Prize, these kind of problems just went away. Yeah.
But new problems appear.
Yes, there's always, I think the to-do list and the problem list is never done.
Yes.
There's always new. It's just a matter of how you approach them, and you move through those steps.
Sure.
So you also mentioned, the one question I always ask, and you kind of alluded to it, is how did you get into studying GPCRs? When you started working on glutamate, did you know that it was a GPCR ligand? And what was that first moment, or do you remember the first moment when you realized actually we're working with the GPCR?
When was it? It's an interesting story. Again, different people working on different topics in the same lab and talking to each other. This is the trick. I mean, the early 80s was the period when people realized that GPCR were not only producing or limiting the production of cyclic AMP, was the point where people realized that IP3 was also a second messenger. And so people were started to study IP3 production in different cell types and putting different ligands and discovering new pathways for GPCR. And this was at that period. And just on the neighboring laboratory, Fritz Ladecek and Samuel Weiss, two scientists in Joel Bockert's lab, were studying the alpha-1 adrenergic receptor. And of course, Chris Kirk, probably most of you don't know him, but he was at the origin of the discovery of IP3, came and did a sabbatical in our lab and bring all the technology to measure IP3 production. At that time was kind of difficult with these big columns, even HPLC, to separate the different inositol phosphates. And Fritz Sladecek did all these experiments on the alpha adrenergic receptor and discovered that the alpha-1 were indeed coupled to PLC and producing IP3. So because we had this glutamate receptor binding site, not related to ion channels, we knew that we didn't have any effect on producing cyclic AMP neurons, was not working well. So we said, why don't we give it a try? And that's how we jumped into discovering that glutamate and quisqualate or other glutamate analogs could produce IP3 in culture neurons with a pharmacology that was not very similar to the other ionos covered this, and then metabotropic glutamate receptors.
Wow.
And then, if we can go along these lines, I mean, when you make such a very important discovery, it was thanks to the association between different people. One believing that IP3 is a second messenger, another one believing that some excited amino acid could be a transmitter, another one doing glutamate binding, and so on. But when you do such a discovery, what would be a dream is that then people, wow, there are so many things to do. We should work together, and you do this, you do that, and so on. But the problem is, human are not like this.
Human
and what
egos.
happened is that a fight started between the people, and I was kicked away and saying, well, you should not work on metabotropic glutamate receptors. So I switched to ionotropic receptors for a while, came back to mGluRs later, due to, egos and was something that, was not really something I really appreciate in that period of my life, scientific life. But in a way, this is happening so often.
Yeah. You make a good point. It's an unfortunate reality that it does happen often. I've heard horror stories.
Major discovery can lead to disagreement between people because who is the father? Everyone want to be the father and want to be the leader and so on. So. Who cares?
Exactly. But, I think over time I learned that you can only work with people who actually want to work with you. And if it's not, I feel like, looking for a job or finding collaborators, and anytime it involves people, it's kind of like dating. You really want to make sure that you have a good foundation, you have open communication, and you have the desire to achieve something together.
Yeah.
And if it doesn't happen, it doesn't happen. It's okay.
Yeah.
But
go on your way, continue what you love. And if you love and express, that your enthusiasm in what you're doing, people will appreciate and will come to you.
Yes. And it's going to change. I mean, the best example I can think of is the podcast. When I initially decided to do the podcast, I reached out to 20 people. Some I knew very well, some I didn't know very well. And I said, this is my idea. I want to shine a light on you. What do you think? 95% responded, 75% said, yeah, great. 25% said, I don't even know what a podcast is. I can read a paper, which is fine, but it's funny because some of those people who said, no, I'm not interested. No, I don't know what you're talking about, ended up coming and talking to me on the podcast. But these people needed time. And, it's what they call, the first one who came were the early adopters, who said, sure, why not? And then afterwards, when you establish yourself and as you mentioned, keep at it, people will realize, oh, actually, this is quite a good idea because it's already established, which is fine.
And it's useful for many people.
Exactly, which is fine. Yeah. All right, let's talk a little bit about more mGluR receptors. Do you have a favorite one?
Heterodimers.
Okay. You're the first one to say that. So I've had answers as to, I don't have a favorite GPCR. I have a favorite G protein. And we took it to the next level, I think, with Andrew Tobin the other day. He said he has a favorite phosphorylation motif, which I thought was really a fun. But you're the first one who comes up with the heterodimer. So which one is it?
Not only one, but I mean, the point is that for so many years, these class C GPCRs were recognized as dimers, that's for sure.
Yes.
And among the class C GPCRs, only the mGlu receptors and the calcium sensing receptor are recognized as homodimers. And this was a dogma. Everybody were convinced that these are and can only be homodimers made of two identical subunits. And it took me maybe 3 years to convince the student to do the experiment and test whether these receptors could, form heterodimeric entities. And because they are covalently linked, these heterodimers may be stable over time and may have specific properties. And this was by around 2010. And when we realized that this does work, at least in transfected cells, of course, the next question was, are they real? When I say real, do they really exist in native systems? And this has been quite challenging. several groups in the world are now working on it, and several ways can be used to validate this. One way is to identify what we could call a pharmacological signature, that you have a specific pharmacological properties that you can reproduce in brain slices, in our case. This is what Colin Niswender and Jeff Cohn did, the same with Jonathan Javic, and the same for us. We did it later than Colin, but at least it brings more data. And the last story we did was just to use antibodies, indeed nanobodies, to indeed demonstrate that we can not only detect the proximity between an NG2 nanobody and an NG4 nanobody in brain slices, in brain samples, but we can also use these nanobodies to develop biosensors. So if you have a nanobody that recognizes the receptor only when being activated, another one that binds the receptor whatever state, you can get threat between the two nanobodies only when the receptor is activated. With this, you can check the pharmacological properties of your heterodimer in real situation with the cells dissociated from the brain. And this is what we did, and we characterized the 2,4 heterodimer, we characterized its pharmacological properties. And surprisingly, we found that in most brain regions, the 2,4 heterodimer is much more abundant than the 4,4 homodimer. So this was a big surprise. I'm very excited about this new finding that we did recently. And fascinating, I think, because was not expecting. And the good thing is that in the field, nowadays, all the pharma industry realized that these things exist, and this totally changed the way they can analyze their molecule. For example, a positive allosteric modulator on a homodimer can become a negative allosteric modulator on an heterodimer. So yes, nothing can be predicted. Everything has to be reanalyzed, looking at these new heterodynamic entities.
I think it's fascinating, and just wanted to mention that I did read your earlier papers, about the heterodimer, and I always thought that they were elegant papers. I read them and I was like, wow, what an elegant way to show that in transfected cells. How cool is this? Not only from a biological perspective, but from also a scientific perspective as to the approach on how to develop methods to actually look at heterodimers in transfected cells. I always thought there was such an elegant way of proving something.
That's something, I always explain to my students and postdocs, and also Philippe Rondard, my colleagues, with whom I've been working for more than 20 years. If we try to do what the others are doing, we're never, ever going to be competitive for a very simple reason. We don't have money in France. If we apply for a grant, the best we can get is 200,000 euros for 5 years. So you can realize that that's really not much compared to, when I review grant from the NIH or whatever, it's, $1 million, not 1/5 of it. So because we don't have much money, we have to do something different. If we want to compete with the people who have the money, we're never going to be successful because we're not going to be fast enough. And so I always said, we have to think, with des bouts de ficelle, as we said in French, with few tricks. And try to develop your own approach to answer the question that you really want to tackle. And this has been something that I tried for many years with the quality control system adapted from the GABA B receptor to other receptors in order to control the subunit composition of dimers. Yeah. This was a good trick that we're still using nowadays. The snap tag and all this technology that we carry out with together with SysBio and the nanobody tools that we develop now, photo switchable ligands also, always collaboration with chemists. I think it's very important to collaborate with chemists. Not always easy, but very, very important. They
Yes,
can provide you tools, and you can provide them. I mean, a collaboration, it has to be a win-win situation. And with chemists, I mean, the chemist has to be happy about the data you provide and the characterization of his or her molecule, and vice versa, his or her molecule has to be useful for your project.
Yeah,
something important in collaboration.
Sorry. No, there was a slight lag between our Zoom, so that's why I don't mean to interrupt you, but I always thought that yes, your work has always been very elegant, very, the tools and all of these things, and it's a small world because I've actually at the Keystone meeting, Philippe Rondard was there, and I've recently met Laurent Prézot, and actually. Karine Monnier, who was in Philippe Rondard's lab, was a colleague of mine. She was doing her postdoc at Rockefeller University at the same time. So I think I even got, while I was at Rockefeller, some cells from Philippe's lab. So it's a really small world. And collaboration is very important. And you mentioned chemists. And I think chemists are fantastic collaborators. And yes, we don't speak the same language, but there is potential in doing some great work with chemists and other people in different areas in order to really be creative and solve problems.
Yeah. So well, we can switch to what is a collaboration, because I think this is essential to be successful. You have to collaborate.
Yes.
Because you don't know everything. You don't have the know-how in every kind of technology. And the more you collaborate, the more chance you have to be successful. It's always something that I really enjoyed, and I always took care for any collaboration that both the participants on the collaboration gonna win something of the common work. So a chemist has to be recognized for the chemistry he's doing, for the ligand he's been developing, and the biologist has to be able to use these tools to make new discoveries in his biological environment. And also it's the same with the company. I mean, we've been collaborating now for more than 20 years with SysBio, now Perkin-Elmer SysBio. It was a French company, now it's an American one.
And I wanted to also mention SysBio. I not only tried some of their reagents, but I've met many people from SysBio, and I still think they were such an amazing team. I could start name dropping, but I really liked, I liked Francis, the little doll that was created at SysBio, and I think it was a great team, and the tools are amazing.
Yeah, and in our case, it's a clear win-win situation that is recognized on both sides.
Yes.
We provide them with ideas, new ways to go. We validate their tools, and we publish their tools in such a way that they are rapidly, highly visible in the field. Yeah. So they also like that. It proves that the tools are of good quality. But at some point, sometimes I received a comment from a referee, and I'm not going to mention who, but I know who he is. And it was a very good point. He said, wow, your papers are fantastic, but nobody in the world can do these things. Because if we had to buy all the tools to do the experiments, it's going to cost us $10 million or whatever, euros, it's almost the same. And it's true that our collaboration is really successful also because of this, because we have access to tools that nobody else in the world can have access to. And or they can, but it's so expensive that nobody can buy it. But I hope that the fact that these tools are so interesting that other ones will develop similar tools and avoid the patents, or maybe the patents are never forever. Yeah. So at some point, these tools will be accessible to everyone and help people advance science with this.
I think, having used the SysBio tools and you having published and helped, collaborated successfully with SysBio, you set the groundwork already, because when you've worked on a receptor, you've published the data. So if anyone wants to replicate, they don't have to start from scratch. Mm-hmm. And I think that also helps in cutting the costs. of these things. And I think with over time, kind of knowing, how much an IP1 kit versus a cyclic AMP pieces bio kit costs, it's not that expensive. And there is a tremendous amount of literature as to where to start, because you have to figure out how many cells and how long and what's the concentration of the ligands and all of these things. And we're getting there. The other interesting point that you made about collaborating, I think over time, and I think you were kind of a pioneer in that, in collaborating with private companies, which wasn't something frequent back in the day, or at least it wasn't out there or put out there or public knowledge. That has been changing. A lot of labs now collaborate with smaller companies in order to work together and to advance and getting access to these tools, which didn't exist before. So I think we're getting better as scientists in general at opening up our labs and collaborating with companies, which also is important because the deliverables and the goals are different, but the common benefit is really advancing understanding GPCR biology.
Yes, totally true.
Which I think it's such a great thing. Before we move on to talk a little bit more about collaboration and how you approach, I want to touch upon how you approach, the ego part. You mentioned had being pulled away from the mGlu receptor project with the IP3, discovery of the IP3 accumulation assay. In your position today, you mentioned that it's very important for you so that with collaborators, it's always a win-win situation. Egos, humans are the same. How do you approach that today? How do you define the structure of a collaboration today?
So I think what is really important is that whoever is in the group, one has to recognize what the other is bringing to the group. And it is very important to have this kind of discussions. And we have routinely this discussion with Philippe Rondard, with Eric Trinquet from SysBio, and with my former postdoc in China at Zhenfeng Liu. All four of us are very different. Each of us brings something to the group because we all four are working together. And not always trying to be, I'm the one. I'm not the one. I'm just talking about the work we are all doing together. And this is something that is important. And then you have to respect that everyone can be last author on a paper. And I don't care nowadays. My career is done. I mean, in the French system, I'm never going to have any increase in salary. So whatever I do nowadays, I'm going to be stuck there forever until I retire. So I have nothing to gain. I have nothing to. So the only thing to do is to allow the others to grow up, to grow, to develop, and so on. So that's why right now I'm not team leader. I have no lab anymore. I'm a scientist working in Philippe Rondard. And I think it is important for Philippe also to grow up. And for me, I'm having fun, helping him to grow up and get recognized for the work he's doing, and vice versa, because we work together. The same with Zhenfeng. If you look at most of our recent publication, we are all three co-corresponding authors in most of the papers. One is last one time, and in another paper it's another one. But all this doesn't really matter. We all have to get benefit from the work we've been doing, and that is very essential.
I like that. Which I think, when you think about it, for someone at the stage of your career where you are, I think it's amazing that you just love science and you're just having fun and helping others, which I think this is the way it should be. But when you
yes, it should always be like this. This is what I keep telling the young scientists, who are eager to have their own group, independent. It's fine to have that as a goal. Because if you don't have a goal like this, you never go into science. But this should not be the primary goal. The primary goal should be doing good science. If you do good science and enjoy science, the career will follow. I never, ever met someone who did good science and was not successful. But I did meet a lot of people who were eager in having their independence and group and so on, and at the end spend too much time trying to, work on their network and always say, I'm the best and so on, but not doing that much. And at the end, they were not successful. I mean, love what you do. And usually the chance that you're going to be successful is never 1/1, but close to 1.
Yeah, and I think it's important. You're making a very important point as to really follow the science and love what you do, and the rest will come, because that's what comes through to people, is how much time, how much effort, how much you love it, and that is going to be recognized. Now, I think nowadays, with the limited number of permanent positions, you had mentioned that, at that time you came back at a very young age from the US and you got a permanent position. It's not the case anymore. And I think junior scientists and postdocs have to realize that you can be an amazing scientist. You can love what you do, but it does not have to be in academia. It can be anywhere. And you have to keep an open mind about that.
Don't forget, I mean, because of our collaboration, we have a common laboratory between our academic lab and CISBio. So any people from CISBio, from R&D, can come and work in our lab, and vice versa. We can go and work on their site. It's only 100 kilometers away. and the very nice things for the students and postdocs from my group is that basically most of them, if not all of them, being trained as a PhD student or postdoc after their time in our lab, even though they published very nice papers, started to have nice findings and so on, most of them choose to go into the industry. Why? Much less stressful in many cases, much better organization, the responsibilities are clear, and no such an issue with grant system. So it may be specific to the French area where it's kind of difficult to raise money to do good science. But at least most of my former students and postdocs are in industry and quite successful.
Yeah, I think you make a good point about, not having to think about fundraising, and you don't have to think about those. And I think in industry, especially now, from what I'm seeing, you have clear goals, clear deliverables, and a lot of times academics used to say, well, in industry you cannot be creative, or you cannot, and it's not true anymore. You can be creative, you can have a portion of your time dedicated to a special project. Who knows? Maybe you're going to discover something and you're going to shift the entire, pipeline of the company through that discovery. And not to, say don't go into academia, but it's difficult. And I think nowadays we have to realize that if your goal is to do good science and enjoy what you're doing, it kind of doesn't matter where you do it, as long as you're really good at what you're doing and you really enjoy it.
And if you enjoy it, you can still, what is good in academia, if you really want to be successful, you have to narrow it down, talk about the amino acid number 347 and concentrate on the movement. The more focused you are, the better chance of success, in a way. I remember a friend of mine saying there are 7 rules to be successful in science.
Okay.
The first is focus. The second is focus. The third is focus, and so on, up to focus at the end, which is true. And at the end, it's frustrating. If you most people are so much concentrated to be successful in their field that at the end they are not so much open-minded, and they are sometimes a very limited view of science. Whereas if you go into industry, because you change topic, you have to. People are much more open-minded because they have to hear, they know much more about different topics, different fields. And so intellectually, it is also very exciting.
I think so too. And I think you make a great point about focusing. And I listen to a lot of talks, and sometimes I disconnect because that person is presenting, as you mentioned, about that amino acid or that group or that phosphorylation site, and it's so focused, and they have a hard time putting the presentation in a format that anyone with a scientific background might be interested and might understand. And if you don't understand the lingo, you're like, okay, so
who cares? Who cares?
Exactly. That's and it's interesting because for a long time in my mind I thought, I may have not been smart enough to understand this, but now with some recul, I realized that it's not me, it's that focus.
Yes.
And you can't be an expert at everything, but you can be a generalist.
Yes.
With a lot of expertise. I asked this from everyone, and I think the answer is 99.9% of the time is yes. Do you think GPCRs are still good drug targets, and why?
Yes, of course. Otherwise, I wouldn't still work on this. I had that feeling, especially in the glutamate receptor field, that mGluRs will be an excellent target and will be a target used in medicines at the end of my career. Because I started my career participating in the discovery of this. I said, 40 years ahead, there will be a drug. I was wrong. So far, there is no drug on the market targeting these receptors, but I still believe that there are very exciting targets. And for the other GPCRs, obviously, I mean, it's a question of time. It's a question. I mean, if you look at, Fiona Marshall showed me like a few years ago the number of new drugs on the market. Most of them were still targeting GPCRs. This was like a few years ago, and I guess it's still true. I mean, I didn't check this last year what was on the market and how many of these drugs were targeting GPCRs. And I also believe, drug targeting GPCRs may not necessarily only be small molecules.
I agree. There is a revolution with therapeutic antibodies right now. It's the new craziness, and which is a good thing. It's also a challenging problem due to all sorts of technical issues. If there were a drug targeting mGluR receptors, what would that indication be? In what context?
I still do believe schizophrenia is a good target for drug targeting mGluR, especially mGluR2 or maybe mGluR5, or maybe both at the same time. This is my goal right now. I do believe 4 can be a good target for Parkinson's disease, but one has to take into account these possible heterodimers. And that's an issue that both Colin in Jeff Cohn's lab and our group are taking into account nowadays. But these two targets I do believe they're going to make it one day. I bet.
Okay. Well, the minute it happens, we have to jump on another podcast episode too to discuss this. What do you think is missing from the mGluR field, or when it comes to potentially methodologies or key information in order to get to that drug? What are we missing?
So far, the drug companies the first molecule who went on clinical trials were small molecules acting as agonists. And these small molecules acting as agonists do not go into the brain very easily, so you had to use prodrugs. They were not selective. They were targeting several GPCRs at the same time, mGlu2 and mGlu3 in that case. So these were not necessarily the best drugs possible to go for a therapeutic application. The second series of drugs, and the fascinating drugs, are the allosteric modules. And these are, of course, when these have been discovered in the early 2000s, they were like new field open, new possibilities, wow, fantastic, and so on. But right after, a few years after, I said, well, if you look at the TM domains of mGluRs, there is no sodium site, there is nothing at all polar. So any molecule binding into this allosteric site in the mGlu receptor within the 7TM domains are all highly hydrophobic molecules. Not necessarily the best way to develop a drug. It's very good because it goes into the brain, but it sticks everywhere, and any protein with an hydrophobic core has an hydrophobic pocket where these hydrophobic molecules could fit. So the possibility of off-target of these allosteric molecules targeting the 7TM region of mGluRs is high. And this is, in my view, a limitation for these molecules. But, then it's luck.
Yes.
one can find a drug that can make it and go up to the market. But yeah.
Hopefully soon, sooner than
So
later.
let's go. Maybe nanobodies will be a new way to go. Who knows?
Who knows? And I think, there's advantages and disadvantages to small molecules versus antibodies, nanobodies, and maybe we'll find that combination. Even antibody drug conjugates could be that middle road.
I have a grant on that specific issue, having a nanobody on one subunit and then a ligand attached to the other subunit in order to control the heterodynamic entities. So this is also an interesting project.
Awesome. All right. Last but not least, we've talked a little bit about advice and to how to contribute to the field, about the fact that you need to love science, you need to love what you do, and the rest will kind of come. What other advice would you have for young scientists who want to contribute to the GPCR field in general, potentially for the into the Class C mGluR receptor field?
Maybe start doing something else than GPCRs and bring the technology to the GPCR field. I think we have to learn a lot from physicists, for example, from mathematicians, from all the fancy microscopy that are being developed nowadays. So I think I would recommend the young people to go into this, learn about fancy technology that are not yet being commonly used in the field, and then apply that to the field. That is a way to go. It's one possibility, at least. The first I think of.
I think that's a great point, because other fields are doing the same, coming into the GPCR field, learning about what we've been using as methodologies. I'm thinking Fred, Brett, you name it, and then applying it to some other field in order to understand, conformation functional relationships of their favorite proteins. Yeah, which it goes both ways. The door swings both ways in that sense. All right. Again, I have two more questions for you. We've talked a little bit about, how you got into the field and how you ended up working on mGluR receptors. Any aha moments that you had during your time as a scientist that shaped your trajectory? It could be also a personal experience that without which you think you'd be a different person today.
So I already mentioned this situation when we discovered the mGluRs and the split between the people instead of working together. This has been something that affected me as a scientist. But then I came back to the field with another perspective. Then what else?
While you think about it, I think that you had mentioned going to university with the idea that you wanted to be a teacher, and then you met a scientist at the university and you realized actually doing research might be also a rewarding.
Well, the point is that I met these teachers at the university who teach me about neuropharmacology, so that you can control your mind, your behavior, your brain, basically, with small molecules, was antipsychotics and all anxiolytics and all these molecules. And I said, I want to discover new, and I want to work on the discovery of such molecules for the future. And what is really disappointing is that so far we didn't do much over the last 50 years in that field. So still, that's something that is of much interest to me. And if I can do anything, trying to help companies discovering new ways to treat these patients, that would be really fantastic. But I also realize that drugs may not be the only way of treating psychiatric diseases. And we also have to consider other ways that can improve the quality of life of these patients. And so this is also something that is one has to consider. I don't know.
Okay, perfect. I think the last question, I don't think it matters at this point, because you had mentioned that you don't have a lab officially anymore, but you work in Philippe Rondar's lab and you're helping him. But if there are any job openings in Philippe's lab, or working with you, collaborating with you, where can people find you?
Where
can people find you? Let's say someone is listening to the podcast episode and they say, I have a great idea, a great new technology, I want to work with you. Where can people find you?
Just contact me by email, through LinkedIn. Give me a phone call. I mean,
do people still do that?
I'm very open. I have no more administrative responsibilities. I did that for 15 years, running a big institute. And this has been really difficult to carry at the same time, all the administration and the science. At the end, I decided to do only science. I love it. So my door is open to anyone all over the planet to come and talk to me, come and visit. We had Ali Salapour, probably you know him.
Yes, of course.
He was with us. He's still with us because he's going to go back to Toronto next week. So whoever want to come and practice the technology we're using, come in. We're going to have fun with you. So just ring at my email or whatever.
That's perfect.
And we'll have a chat.
That's perfect.
With pleasure.
Thank you so much, Jean-Philippe, for your time.
Thank you for all you do for the GPCR field, Yanina.
Thank you. I'll talk to you soon.
Bye-bye.
Thank you for joining us and listening in to this Dr. GPCR podcast episode. I'd like to thank our team members, Attila Forrest, Inés Pinheiro, and our newest Dr. GPCR protege, Montserrat Avila Zozoya. Welcome to the team, Monse. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at any time at hello@drgpcr.com. Until next time, stay safe.